Welcome, everyone. I would like to begin by acknowledging the traditional owners and custodians on the land on which we're meeting here today, the Wurundjeri people of the Kulin Nation. I acknowledge their elders past, present, and emerging. I also acknowledge any Indigenous people dialing in online or from other lands with other traditional owners. It's a great pleasure for me to introduce today's Wednesday seminar speaker, Diane Moujalled. So Diane did her PhD at La Trobe University with someone I know fairly well, Hamza Putalakat, and she survived that quite well, which is a credit to her. Maybe also credit to Hamza, I don't know. And she then worked as a postdoc for a while at the Florey Institute on neurodegenerative diseases. And then she came to WEHI to work with Emma Josephson, initially in the Cancer and Hematology Division, and then moved into Jeff and Jane's Stem Cells and Cancer Division when Emma relatively abruptly left Australia, not because of Diane, I think. Warren very strongly encouraged me to take on Diane, and I'm grateful to Warren for doing that, and I certainly never regretted it. So Diane is part of the WEHI's Brain Cancer Center, and her work that she will present to you today is centered on her work about how to use initiators of programmed cell death, different types of programmed cell death, to get improvements in brain cancer therapy. So over to you, Diane.
Okay, thank you, Andres, for that introduction. And so for today's seminar, I'll be giving an update on our research program that forms part of the Pathways to New Brain Cancer Medicines theme of the Brain Cancer Centre, where we're really aiming to explore cell death-based treatments, which we hope will better inform therapeutic strategies in brain cancer. And so despite decades of research, brain tumours remain among the deadliest form of cancer, and patients are faced with a dire prognosis. So over the last 30 years, the number of people surviving other types of cancer has steadily improved. However, the survival for brain cancer remains unchanged. And we're also faced with one Australian being diagnosed with brain cancer every 5 hours, brain cancer killing more Australian children than any other disease, and brain cancer killing more people under 40 than any other type of cancer. And so the tumours can be classified based on a grading system, being ranked from least aggressive, so a grade 1 glioma, to most aggressive, a grade 4 glioma, and this includes glioblastoma, which is a notoriously fast-growing and aggressive brain tumour. So we can see with the higher grade gliomas that the survival of patients is drastically reduced post-diagnosis. However, with early diagnosis, this is linked to an increase in survival times of the patients. So GBM is the most common and aggressive form of brain cancer, accounting for around 45% of malignant tumours diagnosed in adults. And the incidence does increase with age, becoming the most common malignancy of the CNS diagnosed in adults. And frontline therapy involves maximal surgical resection, followed by chemotherapy with the DNA alkylating agent temozolomide and radiotherapy. But despite this multimodal treatment, recurrence is inevitable, and the median survival remains at around 15 months. We can see, compared to other cancers of the CNS, that the 5-year survival for GBM is considerably lower. And these tumours can arise anywhere in the brain. They're usually found in the frontal or the temporal lobe, sometimes can spread to the spine, but generally do not metastasise outside the CNS. And so here is a timeline of FDA-approved treatments for GBM, with survival curves from 3 pivotal randomised controlled clinical trials. And this led to the standard of care treatment in 2005, the STOOP protocol, which involves radiotherapy and temozolomide, followed by adjuvant temozolomide. And then also the addition of the VEGF inhibitor bevacizumab to standard of care, and also the addition of tumour treating fields to standard of care. But we can see, despite the addition of these different therapies to standard of care, the overall survival of patients is very poor, and there really is an urgency to develop new therapies for GBM. So why are these tumours so challenging to treat? There are many factors that contribute to this, and it's partially due to the anatomical location of the tumour. We know that for chemotherapeutic agents to effectively kill these tumours, they need to bypass a formidable obstacle, being the blood-brain barrier. So the blood-brain barrier is made up of various cell types, including astrocytes, endothelial cells, and immune cells, and the blood-brain barrier can exclude around 98% of small molecules from entering the brain. The unique biology of GBM tumours can also give rise to inter- and intra-tumour heterogeneity, and the tumour microenvironment can also play a role. GBM cells can infiltrate deep into the brain, and glioma stem cell populations can also contribute to the high degree of plasticity. There are also major problems with the current standard of care in that temozolomide is poorly soluble, and only 20% can actually reach the brain parenchyma. But despite this, there are emerging therapies which include precision oncology, as well as immunotherapy and also combination therapy, which is the core focus of our research, where we aim to exploit programmed cell death pathways, as well as pyrimidine nucleotide metabolism. So we know that GBM cells can evade programmed cell death signalling, and this contributes to evasion of chemotherapeutic agents. And so one pathway we aim to exploit is the intrinsic pathway of apoptosis, which is regulated by the BCL2 family of pro- and anti-apoptotic proteins. And so we know in healthy cells, the effector proteins BAX and BAC are restrained by pro-survival proteins such as MCL1, BCL2, and BCLXL. And upon diverse stress stimuli such as DNA damage or cytokine deprivation, this can promote the upregulation of BH3-only proteins, which can bind and neutralise the function of pro-survival proteins, unleashing BAX and BAC, and this promotes a cascade of caspase activation and apoptosis. And so there's a class of small molecule drugs known as BH3 mimetics, essentially designed to bind with high affinity to pro-survival proteins, triggering apoptosis. So for our work, we're really interested to use S6345 to inhibit MCL1, venetoclax to inhibit BCL2, and also A1331852 to inhibit BCLXL. And these BH3 mimetics are really emerging as promising therapies for the treatment of hematological and possibly solid cancers, with venetoclax leading the way, FDA approved for the treatment of CLL in 2016. There are also MCL1 inhibitors and BCLXL inhibitors currently in preclinical investigations and in early phase clinical trials. And so unsurprisingly, in GBM, the expression of pro-survival BCL2 family proteins has been shown to be dysregulated. And high levels of BCLXL are associated with rapid progression and poor survival of GBM patients. And so based on this, BCLXL is proposed as a marker of therapy resistance. The downregulation of BCL2 and BCLXL with bispecific antisense treatment was shown to potently kill GBM cell lines, and conversely, the overexpression of BCLXL was shown to promote cell migration and invasion. And so a few years ago now, we examined the therapeutic potential of BH3 mimetics in GBM, and we reported that BH3 mimetic drugs can cooperate with temozolomide, the standard of care agent, as well as the bromodomain inhibitor JQ1, and small molecule inducers of ferroptosis, Erastin and RSL3, in killing GBM cells. And what we showed was that the combination of BCLXL inhibitor with either temozolomide or JQ1 could enhance killing of GBM cells compared to monotherapy treatment of these agents. We also showed that the co-targeting of MCL1 and BCLXL with BH3 mimetics really exerted potent anti-proliferative effects in 6 human GBM cell lines. So this combination was more potent compared to treatments with temozolomide monotherapy. And so following our study, another group then reported a few months later that there's increased apoptotic sensitivity in GBM cells, and this enables therapeutic targeting in BH3 mimetics. And what they showed was in 3 different patients that the expression of BCLXL and MCL1 was more abundant in the tumour core relative to the non-tumour margin. And the gene expression of BCLXL and MCL1 was also significantly higher in the higher-grade gliomas compared to normal brain tissue. With their in vivo studies, they also demonstrated that in mice intracranially injected with patient-derived glioma stem cells and with MCL1 deleted by CRISPR-Cas9, this augmented the tumour growth, and this was also reflected by prolonged survival in the mice. And so together, their study showed that MCL1 does play a key role in GBM tumorigenesis in vivo. And so apart from dysregulated pro-survival BCL2 family proteins in GBM, there are also alterations in metabolic pathways that can act as drivers for GBM progression. And this is shown to be a multifaceted process and really a hallmark feature in GBM. And these alterations include lipid metabolism, as well as in amino acids and pyrimidine nucleotide metabolism. And so just touching on pyrimidine nucleotide metabolism, it is really essential for the production of various macromolecules in a cell, such as DNA, RNA, and glycoproteins. And the salvage pathway is really the main source of pyrimidines for resting or fully differentiated cells, whereas the de novo pathway is the main source of pyrimidines for highly proliferating cells, such as tumour cells, in order to meet the boosted requirement for pyrimidine synthesis. And it's also been previously shown that pyrimidine nucleotide remodeling can also facilitate GBM tumour progression. And so this shift in metabolism in the GBM tumour relative to non-tumour tissue really does present us with a therapeutic opportunity to selectively target the metabolic vulnerabilities in GBM while mitigating any potential toxicity in healthy tissue. And one promising candidate includes DARH orotate dehydrogenase. So DHODH has recently gained some considerable attention as it's reported to be a druggable target in brain cancer. This enzyme is located on the inner mitochondrial membrane and it mediates the oxidation of DARH orotate to orotate, and it plays a key role in de novo pyrimidine synthesis. And so Bayer developed the compound BAY240-234, which antagonizes DHODH and therefore inhibits de novo pyrimidine synthesis. BAY is a neutral small molecule and it's also reported to be brain penetrant, orally bioavailable, and has low blood clearance with a half-life of around 4 hours in vitro. This compound also displayed monotherapy efficacy in PDX models of glioma and in medulloblastoma, which is a pediatric brain cancer. And so our overall research strategy to evaluate the impact of various drug treatments in GBM include employing GBM cell models. This includes human GBM cell lines, as well as patient-derived neurospheres. And in collaboration with the new brain cancer models theme of the Brain Cancer Centre, Claire Story is generating patient-derived brain organoids, also from resected tumours. And so the advantage of using the patient-derived organoids and neurospheres is that they recapitulate key features of the parent tumour, and so they're clinically relevant 3D models to evaluate our drug treatments. And the drug treatments we have an interest in is temozolomide, as well as small molecule inducers of ferroptosis, BH3 medics, and also BAY240-234. And following our in vitro analyses, we also aim to determine the drug tolerability and distribution in vivo. And also with promising candidates, we collaborate with the blood-brain barrier theme of the Brain Cancer Centre, led by Guillaume, and Zung and Gabby are generating a new derivative of an anti-cancer agent in order to evaluate its impact in GBM. And overall, our studies really culminate on evaluating the efficacy of our drug treatments in vivo preclinical models of GBM. So I'll begin now by going through the work that we're doing in patient-derived neurospheres and our in vitro analyses of our drug treatments. And so PDNs, they're really a heterogeneous population of glioma stem cells, and they maintain the gene expression profile and the cell lineage and diversity of the parent tumour. We currently have 3 different PDNs from our collaborator Samuel from the University of Texas Southwestern. And these are images of how these neurospheres look in cell culture, and they're designated 63, 71, or 5 that I'll refer to throughout the talk. And these neurospheres harbour mutations in genes that are frequently mutated in GBM, including EGFR, RB1, TP53, and also in PTEN. And so it's thought that GBM can arise from neural and glial cell precursors, and consistent with that, we see that PDN63 is positive for SOX2, which is a stem cell marker, and also for TUB3, which is a marker of early neuronal cell differentiation. And we began by examining the expression of SOX2, GFAP, pro-survival proteins, and DHODH in our PDNs, as well as human astrocytes. We do see that the PDNs are positive for SOX2, which is more abundant compared to the astrocytes, and the astrocytes are positive for the glial cell lineage marker, GFAP. Overall, for the pro-survival proteins, the expression was quite variable, but noting also that MCL1 and BCLXL were expressed in the human astrocytes and more abundant compared to BCL2. And overall, DHODH expression was more abundant in the PDNs and attenuated in the human astrocytes. And so given the expression of MCL1 and BCLXL in the human astrocytes, it isn't that surprising given that these proteins do play a key role in development of the central nervous system, and high levels of BCLXL are shown to be maintained throughout development, and this is seen well into adulthood. And so given our interest with BAY 240 to 234, I want to touch on the pyrimidine nucleotide synthesis pathway and how we can demonstrate the on-target activity of our compound in our patient-derived neurospheres. And so pyrimidine nucleotide synthesis can occur through the de novo pathway, where nucleotides are generated from amino acids or glucose, or through the salvage pathway, where nucleosides are recovered from degraded RNA or DNA, or uridine acquired from the extracellular space can drive pyrimidine nucleotide salvage. And so here we treated PDN63 with either BAY alone up to 200 nanomolar or in the presence of uridine at 100 micromolar, and evaluated cell viability following staining with Nexin-5 and DAPI following 72-hour treatments. We do see that there's significant killing in response to BAY alone, and when we co-treat with uridine, we see the cells are rescued from pyrimidine nucleotide starvation-induced cell death. And so indeed we do show that there is on-target activity of our compound as the cell death is rescued by stimulating pyrimidine salvage pathway with supraphysiological uridine. So given that we previously reported that combinations of BH3 mimetics exerted potent effects at killing human GBM cell lines, we wanted to determine their impact on our patient-derived neurospheres. And so for these experiments, we treated PDN63 with BH3 mimetics as single agents or in combinations and assessed viability after 48 hours. We do see PDN63 is sensitive to the MCL1 inhibitor as a single agent. And when we co-treat MCL1 inhibitor with BCLXL inhibitor or venetoclax, we can see these combinations are very potent at killing the PDNs. In PDN71 and 5, we also do see a similar pattern of sensitivity to these BH3 mimetics. And when we look at the extent of apoptosis by Western blotting, we do see, following overnight treatments with the combinations of these BH3 mimetics, abundant cleavage of caspase-3 and also accumulation of its substrate cleaved PARP1, so efficient activation of apoptosis. We then wanted to determine the impact of co-targeting DHODH, as well as pro-survival BCL2 families in family proteins in GBM. And for these experiments, we treated PDN63 with either BAY as a single agent or in combinations with BH3 mimetics and also single agents of BH3 mimetics. We do see that PDN63 is sensitive to BAY alone, and when we co-treat BAY with the BCLXL or the MCL1 inhibitor, this killing is enhanced. And what we found for PDN71 and PDN5, that they were quite resistant to treatments with BAY as a single agent. PDN71 displayed modest killing following treatments with the BCLXL inhibitor. And we do see the combination of BAY with the BCLXL inhibitor that the killing is enhanced. And so these combination treatments can overcome resistance to the single agent treatments of BAY or the BCLXL inhibitor. And so we were then interested to determine the role of the effector proteins BAX and BAC in mediating cell death following pyrimidine nucleotide starvation and also in response to BH3 mimetics. And so here, PDN63 cells were generated we knocked out BAX and BAC by CRISPR-Cas9, and we generated single knockout cells, so here, and also double knockout cells, and confirmed loss of these proteins by Western blotting. We then carried out cell death assays in these 4 different lines following 48-hour treatments with BH3 mimetics as single agents, and we do see that the control cells are sensitized to MCL1 inhibitor-induced apoptosis, where we see significant rescue in the single knockouts and back-to-back double knockouts. Following combination treatments, we do see, again, parental cells are really sensitized to cell death, and we do see significant rescue in our back-to-back double knockouts. However, single knockouts for BAX and BAC were still quite sensitive to these treatments. We also examined the viability of these 4 different lines following treatments with BAY as a single agent or with BH3 mimetics. And so focusing here with treatments with BAY alone, we do see across these 4 lines that they are a similar pattern of sensitivity to DHODH inhibition. And this is also evident in response to combinations with the BCLXL inhibitor and venetoclax, And also we do see the combination of BAE with MCL1 inhibitor is most potent, and some rescue in our single knockout and back-to-back double knockouts. But given that the double knockouts were still quite sensitive to treatments with DHODH inhibitor and BH3 emetics, this suggested that perhaps there is another factor mediating this killing. And also, given that the combination of BAE with MCL1 inhibitor was most potent, we then wanted to assess the morphology of these cells. And so we collaborated with Niall from the imaging department here at Lehigh to look at the morphology of these PDNs by confocal live cell imaging. On the upper panel, we've got the control cells or back-to-back double knockout cells treated with indicated agents, either single treatments or in combination. These cells were stained with annexin 5 in cyan or SPI650, a cell-permeable nucleus stain in magenta. And these are representative still images after 72-hour treatments. We can see in the control cells that there's hallmark features of apoptosis. A number of cells are annexin 5 positive, and the cells display nuclear condensation and cell shrinkage. Whereas generally in the back-to-back double knockouts, they were refractory to these treatments. And here are time-lapse movies capturing the final 8 hours of our treatments in control or back-to-back double knockout cells. And we do see in response to vehicle control, essentially the cells, they're relatively happy in culture and they can sustain and maintain these neurospheres over time. Whereas in response to treatments with BAE and the MCL1 inhibitor in our control cells, we do see again the characteristic features of cells undergoing apoptosis. Whereas in the back-to-back double knockouts, there is some death, but to a lesser degree compared to the control cells. And so given also that we did see in our cell death assays that in response to BAE as a single agent, or when combined with BH3 emetics, we do see significant killing in our back-to-back double knockouts, this suggested that perhaps there is another mediator of cell death. And so one candidate was the BCL2-related ovarian killer protein, also known as BOC. So BOC is an effector protein thought to be structurally and functionally redundant to BAX and BAC. And it's also been shown to regulate mitochondrial permeability transition during apoptosis. Unlike BAX and BAC, BOC is not restrained by pro-survival proteins, but its protein stability is negatively regulated by the endoplasmic reticulum-associated degradation pathway. BOC has also previously been reported to be a positive regulator of uridine biosynthesis and also shown to bind to the enzyme uridine monophosphate synthetase, increasing its enzymatic activity. However, colorectal cancer cell lines deficient in BOC were shown to be refractory to 5-FU-induced cell death. And so this link of BOC with uridine metabolism prompted us to examine the expression of BOC in our different cell lines. And so we looked at the expression of BOC in our PDNs as well as in human astrocytes. And we do see that BOC was quite abundant in 1 out of the 3 PDNs and attenuated expression in our non-malignant human astrocytes. BAC and BACs were readily detected across our different cell lines. So in PDN63, we then generated cells that were deficient in BOC, BAC, and BACs by CRISPR-Cas9, and lots of these proteins confirmed here by Western blotting. And as a reminder here, we've got the cell death assays in back-to-back double knockouts, where following 72-hour treatments, we do see that there's significant killing in response to BAE as a single agent or combined with BH3 emetics. And when we perform these cell death assays in our triple knockouts, indeed we do see that these cells are more resistant to BAE and also treatments in response to BH3 emetics combined with BAE. And so this supports a role for BOC in also mediating cell death in response to pyrimidine nucleotide starvation and also BH3 emetic-induced apoptosis. So I've gone through some of the data that we've generated in patient-derived neurospheres. I now want to move on to our other model, which is patient-derived brain organoids in collaboration with the new brain cancer models theme. And so Claire Story has generated some GBM organoids, and she uses a published method where from a resected tumour, within 4 weeks, she can generate these organoids that are grown in a matrigel, and they're shown to maintain the key histological features of the parent tumour. And the downstream experiments that we're interested in include IncuSight imaging, to determine the impact of our treatments on cell invasion and spheroid area, as well as cell titer glow assays and immunohistochemistry. But for the purpose of today's talk, I'll just go through the work that Claire has carried out with InQSI imaging and also cell titer glow assays. So this is some InQSI imaging data in one of the organoids treated with these indicated agents, and staurosporin was used as a positive control. And we can see over a 4-day treatment period that the co-targeting of BCLXL and MCL1 with BH3 mimetics was more potent at reducing the number of invading cells. And this was also reflected by a reduction in the spheroid area of organoids compared to single-agent treatment with temozolomide or the combination of temozolomide with the BCLXL inhibitor. These are some representative images through InQSI imaging again after 3-day treatments, where we do see, in response to the combination of the BCLXL and MCL1 inhibitor, attenuated invasive protrusions from these organoids compared to single-agent treatment of the BH3 mimetics or temozolomide, or the combination of temozolomide with BCLXL inhibitor. And Claire has also been performing some experiments to assess the impact of BAE treatments with BH3 mimetics. And we do again see after 4-day treatments that the combination of BAE with the BCLXL inhibitor could significantly reduce the number of invading cells. Again, this is reflected by a reduction in the spheroid area of the organoids. And by 7 days, with the cell titer glow assays, you can see that the cell viability is significantly reduced following the co-treatment of BAE with the BCLXL or the MCL1 inhibitor. And so Claire is performing a number of treatments in different organoids, and we're aiming to expand our resected tumour specimen panels so we can generate more organoids and also corresponding neurospheres to evaluate the impact of our different treatments. And to now go through some work that we're doing in determining the tolerability of the BCLXL inhibitor as well as BAE in immune deficient and proficient mice. And so we began by determining if the BCLXL inhibitor could penetrate the brain in healthy mice with an intact blood-brain barrier. And for these experiments, NSG mice were treated with either vehicle or the BCLXL inhibitor at 25 mg/kg. And 1 and 4 hours post-drug treatment, we collected the plasma and the brain. And then UPLC analysis was performed in order to determine the abundance of the BCLXL inhibitor in these samples. And so based on the brain to plasma ratio, we found that the BCLXL inhibitor did not penetrate the brain to significant concentrations. And so in healthy mice with intact blood-brain barrier, there is restricted access of the BH3 mimetic. And so we then wanted to determine if the BCLXL inhibitor could penetrate the brain in an orthotopic model of GBM. And for these experiments, we collaborated with Misty Jenkins, Ryan Cross, and Melinda Iliopoulos. And so Ryan and Melinda performed intracranial injections in anaesthetized NSG mice. The mice were injected with Hu251 cells harbouring an M. cherry luciferase reporter. And following engraftment, the mice were treated with either vehicle or the BCLXL inhibitor at 50 mg/kg for 5 days. So consistent with BCLXL inhibition in platelets, we do see the mice exhibit thrombocytopenia, a significant drop in platelet counts. We also collected the plasma, the liver, the spleen, and the brains in order to determine the abundance of the BCLXL inhibitor in these samples. And what we found was that the BCLXL inhibitor was the least exposed organ to the brain, was the least exposed organ to the BCLXL inhibitor compared to the plasma, the liver, and the spleen. But despite this, the BCLXL inhibitor was detected in both tumour and healthy brain regions, and the concentrations were above the binding affinity for BCLXL, which is reported to be in the low nanomolar range. And so with previous studies, BAE has been reported to be well tolerated in various PDX models of glioma, medulloblastoma, and acute myeloid leukemia, up to doses of 10 mg/kg. And in collaboration with the Brain Cancer Research Lab, we wanted to determine the tolerability of BAE in NSG as well as C57 Black 6 mice. And so Shannon Oliver treated these mice with either vehicle or with BAE at 5 mg/kg for 10 days, and also monitored the body weights, where we see across our treatment cohorts, body weights were quite similar. We then collected different organs and plasma to determine the abundance of BAE in our various samples through Metabolomics Australia. Okay, so this is heat map clustering showing the top 25 metabolites contributing to biological differences in our samples. And so through this, we can determine the on-target activity of our compound based on the abundance of various intermediates. And so we can look at the abundance of intermediates that are upstream to DHODH or downstream to DHODH, and based on these ratios, we can determine the activity of BAE in our different samples. And so when we look at the ratio of orotate to carbamoyl aspartate, we do see that there's a significant reduction in BAE-treated brains in both NSG and black 6 mice, and so on-target activity of our compound. We also looked at the abundance of BAE in our brain and plasma samples, and we do see, consistent with previous reports, that BAE is detected in the brain. However, it is significantly higher in the plasma, and this does show that there still is restricted access of BAE into the brain in healthy mice with an intact blood-brain barrier. We were then interested to determine the impact of BAE treatments in vivo on the blood phenotype. And so for these experiments, NSG mice were treated with either vehicle or BAE at 5 mg/kg for 3 consecutive days, and also mandible bled during this treatment schedule, and body weights monitored, where we do see they were quite similar across our treatment cohorts. We do see that 2 days post-drug treatment that the mice exhibit thrombocytopenia, a significant drop in platelet counts, and then a rebound effect where 9 days post-drug treatment we do see higher platelet counts, and by day 13, post-drug treatment we do see platelet counts are restored back to baseline. There was a transient drop in red blood cell counts during this treatment, and there wasn't really any significant impact on white blood cells. And so now we were dealing with 2 drugs that promote thrombocytopenia, and so we wanted to determine if there was a possible sequential treatment window that we could actually apply in our orthotopic models of GBM. And so NSG mice were treated in week 1 with 5 mg/kg of BAE for 3 consecutive days, or in week 2 with the BSLXL inhibitor at 50 mg/kg, again for 3 consecutive days. And during the treatment schedule, animals were mandible bled and also body weights monitored. So we do see that body weights were quite similar across our treatment cohorts. And again, the mice exhibit thrombocytopenia 2 days post-treatment with BAE. Following 2 days post-treatment with the BSLXL inhibitor, the mice exhibit higher platelet counts. And what we found by day 23 of this treatment schedule, the platelet counts remained quite high. There wasn't really any significant impact on red blood cells during this treatment schedule. However, the mice did display leukopenia with a drop in white blood cell counts 2 days post-drug treatment. And by 2 days post-treatments with the BSLXL inhibitor, the white blood cell counts were significantly higher. And by the end of our treatment, the white blood cell counts were restored back to baseline. And so despite the impact of these drug treatments on platelets and white blood cells, we have identified a sequential treatment window that is tolerated in NSG mice that we can carry forward in our orthotopic models of GBM. And so for the next part of the talk, I will go through very preliminary pilot studies that we've carried out in orthotopic models of GBM. And so we've adopted this established method from Misty Jenkins' lab to perform orthotopic GBM analyses. And so anesthetized NSG mice are placed on a stereotactic frame. And by stereotactic guidance, the mice are intracranially injected with U251 cells, and engraftment is confirmed by IVUS imaging. And following engraftment, the mice were treated with either vehicle or with BAE at 5 mg/kg for 4 weeks, and this occurred between Monday to Friday. And we then harvested the brains for immunohistochemistry analyses. So this is the immunohistochemistry analyses in a vehicle or BAE-treated brain. And so we do see that the extent of cell proliferation through Ki-67 staining was attenuated in a BAE-treated brain compared to our vehicle treatment. However, we did not see any significant cleavage of caspase-3, not detected in our BAE-treated brain. And so we do acknowledge that perhaps this was a very late time point of 4 weeks of drug treatment to see any significant protein accumulation of cleaved caspase-3. And so we repeated the experiment again, however, harvesting the brains 4 days post-drug treatment. And this is a Western blot here showing the expression of the glial cell lineage marker, GFAP, in U251 cells. And this is GFAP immunohistochemistry staining in a vehicle or BAE-treated brain. And we do see that cell proliferation assessed through Ki-67 staining was attenuated in a BAE-treated mouse brain compared to vehicle, and a very small number of cells that are positive for cleaved caspase-3 in a BAE-treated brain compared to vehicle. So these experiments are still ongoing, where we really aim to expand our treatment cohorts, carry out single-agent and combination treatments of BAE with a BCL-XL inhibitor. So to summarise this section so far, I've shown that the combination of MCL1 and BCL-XL inhibitors really is potent at killing GBM cell lines and patient-derived neurospheres compared to single-agent treatment with temozolomide. And the cytotoxic effect of BAE treatments as a single agent or when combined with BH3 mimetics is mediated by BAX, BAC, and also BOC. And patient-derived brain organoids display a similar pattern of sensitivity to MCL1 inhibition and BCL-XL inhibition, and also enhanced cytotoxicity in response to combinations of BAE and BH3 mimetics. With our in vivo studies, I've shown that monotherapy treatment of BAE is well tolerated, and we have identified a sequential treatment window for BAE and a BCL-XL inhibitor that is well tolerated in NSG mice. And so now I'll go through some work that we're doing in collaboration with the blood-brain barrier theme of the Brain Cancer Centre, where we're evaluating the impact of a new derivative of a BCL-XL inhibitor in GBM. And so targeted therapies are proving to be a promising avenue for therapeutic intervention in GBM. And we know that with conventional therapy, temozolomide is poorly soluble. It cannot adequately cross the blood-brain barrier and kill GBM tumours. And so one possible solution is to co-opt the brain's endogenous trafficking system to deliver anti-cancer therapies. And so Zhuang and Gabby are generating nanobody or peptide drug conjugates of the BCL-XL inhibitor. And the idea is to exploit receptor-mediated endocytosis to enhance the delivery of the BCL-XL inhibitor across the blood-brain barrier and improve its tumour targeting properties. And the candidate receptors include LRP1 as well as transferrin receptor 1. And so it's previously been reported that LRP1 is overexpressed in GBM as well as in endothelial cells that line the blood-brain barrier. Angiopep2 peptide is a synthetic peptide around 19 amino acids long and shown to bind to the LRP1 receptor. And the angiopeptide paclitaxel conjugate was shown to enhance the delivery of this chemotherapeutic agent by 50-fold into brain metastases, and this was shown in preclinical and clinical studies. And the transferrin receptor has also been shown to be frequently overexpressed in GBM. So we looked at the expression of these receptors in our different cell models, and we do see that LRP1 was quite abundant in the patient-derived neurospheres, also non-malignant human astrocytes, and 1 out of the 2 medulloblastoma cell lines. And generally for the transferrin receptor, we do see it was more abundant in our brain cancer lines compared to non-malignant astrocytes. And so Zhuang has generated 3 different BCL-XL inhibitor angiopeptide drug conjugates, and so designated either BCL-XL inhibitor with an angiopeptide or conjugated with a linker and an angiopeptide or just a linker. And we examined the impact of these different drug conjugates on the viability of patient-derived neurospheres after 48-hour treatments. And we do see that the combination of these peptides with the MCL1 inhibitor was just as potent as the combination of the parent BH3 mimetic combinations of the BCL-XL inhibitor with the MCL1 inhibitor. What we also found was that the BCL-XL inhibitor conjugated to the angiopeptide was quite potent at killing the PDNs as a single agent compared to the parent BH3 mimetic. But given also that the human astrocytes abundantly expressed the LRP1 receptor, we do see that there's a similar pattern of sensitivity. So these peptides are very potent when combined with MCL1 inhibitor. And we again do see that the single agent treatment of these peptides also can kill these PDNs more so compared to the parent BCL-XL inhibitor. And so these are proof of principle experiments showing that we can conjugate the BCL-XL inhibitor to the angiopeptide, which can kill these GBM and human astrocytes, unfortunately. But Zhuang is looking to modify this BCL-XL inhibitor by changing the drug to peptide ratio and also modifying the linker conjugated to the BCL-xL inhibitor. Okay. And so I really want to finish off by going through our future directions. And basically, for the next phase of the project, we wanted to determine if there is potential to use BH3 mimetics, so the BCL-xL inhibitors, in brain cancer with minimal side effects. And so we really aim to do this with the generation of these new derivatives, so hopefully better improved new derivatives of the BCL-xL inhibitor with better tumour targeting properties, and evaluate the full potential of these BH3 mimetics in our orthotopic models of GBM. We're also expanding the research program to evaluate the impact of BH3 mimetics in neurotrophic tyrosine receptor kinase-driven glioma. And to model this, we currently have 2 patient-derived cell lines from our collaborator Chris, from the Institute of Cancer Research. And we're also collaborating with Guillaume in order to determine the therapeutic potential of WEHI 7326 in GBM. So this compound was generated in Guillaume's lab, and it presents a new class of anti-cancer agents that can promote cell cycle arrest and apoptosis, and it was shown to attenuate tumour burden in a xenograft model of glioblastoma, but noting that this was a subcutaneous model. So we really want to explore the potential of this compound in our more clinically relevant models of GBM and also in our PDX models of GBM. And so with that, I would like to really acknowledge Andreas for being a really supportive and encouraging mentor. Since joining your lab in mid-2020, you've really given me the opportunity to expand the brain cancer research program, and I'm really grateful to have the opportunity to work in your lab. Also want to thank Iman, my RA, for being really hardworking and carrying out great research across the various projects that we have within our research program. And also Lauren Whelan in our lab for training Iman and I in various mouse procedures in order for us to carry out these intracranial injections in mice, and also helping us with our various in vivo studies. Also want to thank various students who have contributed to many projects within our research program, and also members of the Strasser and Kelly lab. Sue Hardy for all your help with my various projects and student recruitment, and also members of our division. Thank you all for your help along the way, and also a number of collaborators listed through the Brain Cancer Centre. I really want to thank you all for these pivotal collaborations that we have, and they really are essential for our research program, and really look forward to working with you all in the coming years, and also acknowledge Sam and Linda for their support and help with our research program. I want to also thank our collaborators at the Florey, Ashley and Adam, who have really assisted us with our ferroptosis studies, and Sam and Khalil for providing us with patient-derived neurospheres, and also Niall for always willing to help us with our in vitro cell imaging experiments. And the core facilities at WEHI, I want to thank staff in bioservices, in particular Stephen, for helping us with our intracranial injections and giving us advice with our in vivo studies, and also the animal techs up on level 4, in particular Ketty, for helping us with our intracranial injections. And also additional staff within bioservices, thank you for your help. And again, the core facilities for the FACS and WEHI platforms, thank you for assisting us with our various projects, and also our consumer advocates, Fiona and Kathy, thank you for your commitment and support of our research program, and also the funding that we've received that supports our work. Thank you.
Thank you very much, Diane, and we've got lots of time for questions, starting in the room, and for those attending online, please feel free to put your questions in the chat box and I'll read them out. Anna.
Very nice work. Do you think that humans and mice have different white blood cell relative compositions? So B cells and granulocytes are not at the same ratio in humans and mice. Do you think it would be worth looking at the white blood cell populations individually rather than as a whole?
Yeah, definitely. We can do that to kind of decipher which actual compartments are actually impacted. So I think that's definitely worth pursuing. And I think, definitely we have to do that because with other FDA-approved DHODH inhibitors, they've been shown in clinical trials to have a mild impact on the white blood cell various compartments, so we'll definitely have to do that.
Daniel.
Wonderful talk, Diane. Thanks for that. I had a question about the BOC deficiency blocking the effect of the BAE compound. I thought that was very interesting. Could you tell us a bit more about this link between BOC and uridine metabolism and why that might be?
Yeah, so I think it varies based on like the cell context, but we know that BOC, it is localized at the ER, and in response to pyrimidine nucleotide starvation, this effector protein seems to be primed to have a role with mediating this killing, and it was shown to enhance the binding to uridine monophosphate synthetase. We want to kind of decipher the role of BOC in our cell model. So we're currently generating BOC single knockout lines to see what the cell viability would be like in response to single agent treatments with BAE and BH3 medics.
Hannah again. Over there.
So you showed us that LPR1 is also strongly expressed in the healthy astrocytes, and but the transferrin receptor was much weaker, at a much weaker expression level. But nevertheless, the angiopeptide is the LPR1 binder, isn't it?
Yes, that's right.
So presumably you've got a plan to switch or to try at least the transferrin receptor binding peptide as essentially as a mechanism of getting more focused on the glioblastoma cell lines.
So Gabby Watson is generating, well, she has, and we have some agents to test, just not in time for this talk. But yeah, we anticipate that we'd get a better response given the expression that it is quite different in the brain cancer lines compared to the astrocytes. So we do have an antibody for targeting the apical domain of the transferrin receptor, conjugated to B-cell XL inhibitor, that we hope will be more positive data with that. Yeah.
Thanks, Diane. My question's about the nanobody conjugates as well. So these receptors, is there expression of these receptors in other tissues, and could you accidentally target the drugs to the wrong compartment by that as well?
We know that the expression of these receptors, it is quite ubiquitous across different cell types, but with transferrin receptor 1, for instance, for the conjugate of B-cell XL inhibitor, it's not expressed on megakaryocytes or platelets, so that's promising that we're not likely to get thrombocytopenia. LRP1 isn't expressed also on the megakaryocyte lineage, but there still is potential systemic toxicity on other cell types. It's probably just a matter of determining the ideal dose to mitigate that.
So there's one question from online, and then Tim will be next. So the question online is, have you tested whether your BCL-XL inhibitor conjugates / nanobodies reduce platelet killing in vivo compared to the parental unconjugated BCL-XL inhibitor?
Not yet. So I think once the generation of these conjugates are optimized with our in vitro studies, then we'll definitely be doing that in vivo. It's a bit premature now with what we currently have, but that's the idea.
Okay.
So the choroplexus makes cerebrospinal fluid, which is derived from neural tissue, and in the choroplexus there is no blood-brain barrier in the blood vessels. The blood-brain barrier is between the epithelial cells of the choroplexus, and it has a huge blood flow through it. So I was just wondering whether or not it's possible that some of these compounds, whether they're conjugated or not, actually tend to accumulate in the choroplexus, and whether, I guess you haven't looked to see specifically in the choroplexus whether that's the case, but otherwise you would have mentioned it. But I'm wondering whether anybody ever looks at the choroplexus for drug accumulation, and whether or not that's actually an area where the drugs are more likely or that potentially less likely to cross the blood-brain barrier.
Yeah, that's a very interesting point. So the way we do it is we're like macro dissecting out the brain, healthy region and tumor region, but most definitely if we can look at region-specific areas to see where the drugs are accumulating, that would obviously be a great indicator of what's actually happening with the drug distribution. So that's a good point.
Thanks, Diane. The heterogeneity that's supposedly sustained in the organoids, that surprises me, and I wonder how that was determined, the heterogeneity matching in the tumour versus the organoid, and how long it lasts?
So with these organoids, they're shown to be grown long-term in cell culture, like for months and months. And when they look at various markers, like neuronal stem cell markers or markers of neuronal cell differentiation, or even markers of endothelial cells, because these organoids do maintain the tissue architecture, they're maintained through like whole genome sequencing and like through histology analyses. And Claire, I believe, has been doing these assays from when she generates like early generation of these organoids and late into culturing in vitro and showing that they are actually similar to the parent tumour.
Did that surprise you?
But they don't...
What does that say about the competition within the organoid? It's almost as if it's non-existent. That just surprised me. That's cool. Very good.
Well, it's good for us.
Great for you.
Remains to thank Diane for a very clear.
Thank you.